Today, we are taking a deep dive into our source material, specifically decoding Chapter 13, which is titled The Staphylococci. And honestly, reading through these sources, it felt less like a biology textbook and more like, I don't know, a military strategy guide for an alien invasion.
Oh, totally. The warfare analogies just write themselves.
Because we're not just talking about germs here, we're talking about a genus of bacteria that ranges from harmless roommates to fatal killers. And that's our mission today, to really understand that dynamic.
And it is a massive paradox, isn't it? That's what stands out most in the literature. These bacteria are ubiquitous. They are part of our normal flora. They're supposed to be there.
They're on everyone listening to this right now.
Everyone. Yet Staphylococcus aureus, the absolute ringleader of the group, is a major pathogen. It causes everything from that minor pimple you popped yesterday, which you shouldn't do, by the way, to life-threatening sepsis and organ failure.
So, our roadmap for this deep dive is to unpack that paradox. We're going to look at what these things actually look like, their morphology. We're going to break down their superpowers, and I'm really not exaggerating. They have literal superpowers like chemical weapons and armor.
Bioarmor.
Yes, bioarmor. And we'll look at their intelligence because apparently they can talk to each other to coordinate attacks. And finally, we have to talk about the terrifying rise of antibiotic resistance, specifically MRSA.
Which is probably the most pressing medical challenge related to this chapter. But before we get to the scary stuff, we really have to understand what we are looking at. We need to visualize the enemy.
Right. So let's start with section 1, the cast of characters. If I'm a microbiologist looking under a microscope, how do I know I'm staring at a staph infection and not, something else?
Well, the name actually gives it away if you know your ancient Greek.
Staphylococci.
Right. Staphyl means a bunch of grapes, and coccus means a grain or a berry. So when you look at them under the microscope, they literally look like grape-like clusters of spherical cells.
Grape-like clusters. That's the classic Biology 101 visual. But I want to pause there for a second because I think we gloss over why they look like grapes. I mean, is it just an aesthetic choice by the bacteria?
Not quite. It all comes down to geometry and physics.
Yeah.
You might know about Streptococci, the screp throat bugs.
Right.
They form chains like a string of pearls. And that happens because they always divide along the same linear plane. It's like slicing a loaf of bread, one slice after another, forming a straight line.
Okay, so strep is linear, like a train.
Exactly. But Staphylococci are chaotic. They divide multiple irregular planes. So one generation splits left-right, the next splits up-down, the next is diagonal. And because they stay attached to each other after dividing, you end up with this three-dimensional clump.
So it's less like a string of pearls and more like a pile of marbles you've just glued together.
Or grapes, hence staphel. And this structure actually matters a lot for their survival. It makes the colony structurally robust. It's much harder for the environment to break up a cluster than it is to snap a single chain.
It's a defensive formation, like a Roman phalanx. Now, regarding their size, the text says they are about 1 micrometer in diameter. That number is really hard to visualize for most of us. How small is that, really?
Think of a single period at the end of a sentence in a printed book. If that period were a red blood cell, which is about 6 to 8 micrometers across a staphylococcus, would be a tiny speck of dust sitting on that period.
Wow.
Yeah, you could fit dozens of them inside a single red blood cell if you packed them in there.
And yet, as we're going to find out, this literal speck of dust can kill a 200-pound human in a matter of days.
That is the terrifying asymmetry of microbiology. Very small things can cause massive systemic damage.
Okay, so now let's talk about the paint job. When we do the famous Gram stain, which for those listening separates bacteria into the purple team and the pink team, where does Staph land?
They are firmly on the purple team. They are gram positive. That means they hold on to that crystal violet stain during the testing process.
Why is that? What makes them hold on to the purple dye?
It's all about the architecture of their cell wall. Gram-positive bacteria have a very thick, mesh-like layer of peptidoglycan on the outside. When you pour the dye in, it just gets physically trapped in that thick mesh.
And the gram-negative ones?
Gram-negative bacteria have a much thinner wall, so the dye washes right out when you rinse it. However, the source text notes an interesting quirk about staph, which is aging cultures.
Like if the bacteria just get old.
Exactly. If the culture sits around and gets old, the cell walls actually start to degrade. They lose that structural integrity, so they lose the ability to hold the stain. They might actually appear gram-negative or pink under the scope.
Ah, so if you're a lab tech, you really need a fresh, healthy culture to get an accurate ID.
You absolutely do. Otherwise, you might misidentify it entirely.
Okay, so we've got gram-positive grapes. Now, are they moving around? Do they have little tails or propellers?
No, they're strictly non-modal. They don't swim.
Yeah.
They don't have flagella.
Yeah.
And they are non-spore forming.
Which means what exactly?
Some bacteria, like the ones that cause anthrax or tetanus, form these virtually indestructible spores to hibernate for centuries in the dirt. Staph doesn't do that. It doesn't need to, because it's incredibly hardy on its own as a vegetative cell.
How hardy are we talking?
Well, the sources emphasize that they are relatively resistant to drying, to heat, and to salt. They can withstand 50 degrees Celsius for 30 minutes.
That's halfway to boiling.
Right. But the salt tolerance is the real superpower here. They can survive in environments with 10% sodium chloride.
Why does it matter so much? Why is salt tolerance such a big deal for a bacterium?
Think about where they live. Your skin.
Sweating.
Exactly. When you sweat, you are releasing salt water onto your skin. Most bacteria would just shrivel up and die in that environment due to osmotic pressure. The salt literally sucks the water right out of their cells.
But Staph just shrugs it off.
It loved it. That's exactly why they are such successful, dominant colonizers of human skin.
It's like they evolved specifically to live in our sweat.
They absolutely did. And we actually use this to our advantage in the laboratory. We use something called mannitol salt agar.
What's that?
It's a petri dish filled with a jelly that has a very high salt concentration. If you take a swab from a random table, you'll pick up lots of different bacteria. But if you rub that swab on that salty plate, most of them will just die.
But the Staph survives.
Right. Only the staph will grow. It's a brilliant way to isolate them from a crowded sample.
That's clever. The environment that kills all the competition is the exact one they thrive in. Now, the text mentions there are at least 45 species of Staphylococcus, but we really only care about the big four in a clinical setting. Who are they?
Right. At the very top of the hierarchy, the gold standard of bad news, is Staphylococcus aureus.
Aureus meaning gold, right? Like Oreo on the periodic table.
Exactly. When you grow them on a solid media in the lab, the colonies often have this deep golden yellow pigment. That pigment is actually a carotenoid, which is chemically similar to what makes carrots orange. And S. aureus is the dangerous one. It's what we call coagulase positive.
We'll definitely get to coagulase in a second because that seems to be the defining line. Who's next in the big four?
Next is the ubiquitous, usually harmless cousin, Staphylococcus epidermidis. The clue is in the name there, epidermis. It lives on the skin.
So it's the white one.
Yeah, its colonies are usually gray or white, not gold. And crucially, it is coagulase negative. For a really long time, medical science thought it was completely harmless, simply a passenger on our bodies.
But that changed.
It did. It has emerged as a major issue for people with artificial implants. Things like artificial heart valves, hip replacements, catheters. It loves to stick to plastic.
Okay, so S. aureus is the golden killer. S. epidermidis is the white skin dweller that attacks cyborg parts. Who else?
There's Staphylococcus lugdunensis. Now, this one is tricky.
How so?
It's coagulase negative, just like epidermidis. But clinically, in the hospital, it behaves like aureus. It can cause very severe destructive infections, similar in severity to golden staph. It's a wolf in sheep's clothing.
And the fourth one?
Staphylococcus saprophyticus. This is a very specific niche player.
Yeah.
It's a key cause of urinary tract infections, particularly in young, sexually active women. It doesn't usually cause big abscesses on the skin, but it loves the urinary tract.
Okay, so those are the four main characters. You mentioned this coagulase test as the defining line. What is that test exactly?
It's the very first question a microbiologist asks once they know they're looking at staph. They ask, is it coagulase positive?
And coagulase is what?
It is an enzyme that clots blood plasma. S. aureus makes it. If you mix a sample of S. aureus with a test tube of liquid plasma, it rapidly forms a solid, jelly-like clot. The other three, the ones we call CoNS, or coagulase-negative Staphylococci, do not do this.
It seems strange that clotting is the main identifier. Is that just a convenient lab trick, or does it actually help the bacteria in some way?
Oh, it helps the bacteria immensely, which we will definitely discuss when we get to their arsenal. But yes, functionally in the lab, it's the primary way to quickly tell the killer S. aureus apart from the milder skin cousins.
And another test the source material mentioned was hemolysis.
Right. S. aureus is usually beta hemolytic.
Meaning it destroys blood.
Exactly. If you grow it on blood agar, which is a petri dish that literally has sheep blood mixed into the jelly S, Aureus releases toxins that burst the red blood cells open.
That sounds aggressive.
It is. You actually see these completely clear halos around the golden colonies where the blood has been completely destroyed and consumed. S. epidermidis, on the other hand, usually doesn't do that. It just sits on top of the blood without destroying it.
It's aggressive from the very start. It's not just sitting there. It's literally dissolving the blood around it. And I think that is a perfect segue to section 2 of our deep dive, the armor. Because these bacteria aren't just naked cells floating around. They are built like microscopic tanks.
A tank is the absolute perfect analogy. And every tank needs a heavy hull. In bacteria, that hull is the peptidoglycan.
Let's break that down. We talked about it trapping the purple dye, but what is it functionally?
This is the thick, rigid exoskeleton. It's a complex polysaccharide structure. Basically, it's long sugar chains that are cross-linked by short peptide bridges. It gives the bacteria its structural shape and, most importantly, protects it from bursting due to its own internal osmotic pressure.
But it's also a beacon, right? The text mentions that peptidoglycan triggers the immune system. It acts like an endotoxin.
Yeah, that's the double-edged sword of the armor. What does it mean when we say it acts like an endotoxin? It means that just the physical chemical presence of that wall material stimulates your immune cells to produce interleukin-1.
And interleukin-1 causes fever, correct?
Exactly. You don't even need the bacteria to be alive for this to happen. If you just took a syringe of dead S. aureus cell walls and injected it into someone, they would still spike a fever and their white blood cells would rush to the scene.
It's like the bacteria is constantly broadcasting, "I am here, come fight me."
Right. It can't hide its hull. Now, connected to this wall are things called teichoic acids.
Let's talk about those.
These are polymers that are chemically linked right into the peptidoglycan. Think of them as the steel rebar reinforcing the concrete of the cell wall. They are highly antigenic.
Meaning your body builds specific antibodies against them?
Exactly. They help the bacteria metabolize and maintain the cell wall structure, but again, they are bright red flags that the immune system easily recognizes.
Okay, so the wall is super strong, but it's also very visible to the microscopic police. But here is where it gets really, really interesting to me. We need to talk about protein A. The source material highlights this as a crucial virulence factor. To me, reading about this, it sounds almost like a judo move.
That is a brilliant analogy. I want to spend some time on protein A because the text calls this a virulence factor, which is just a sterile scientific way of saying weapon. But the mechanism of how it works sounds like martial arts. It's using the opponent's own strength against them.
Explain the mechanism for us. How does it work?
To understand why protein A is so devastating, we first have to look at how your immune system should work in a normal scenario. Normally, your body produces antibodies to fight infections, specifically immunoglobulin G, or IgG.
These are the Y-shaped missiles that patrol your blood, right?
Exactly. Think of the capital letter Y. The two tips at the top of the Y are the grabbers. They have a specific shape designed to lock onto the bacteria's surface, like onto that peptidoglycan, or the teichoic acid we just talked about.
Okay, so the top of the Y attaches to the bug.
Right. And the bottom stem of the Y, the vertical part, is called the FC region. This is essentially the handle.
The handle is for your own white blood cells to grab onto.
Yes. A macrophage, which is a massive eater cell in your immune system, cruises around looking for those specific handles. If it sees a handle sticking out, it grabs it and eats whatever is attached to the other end. The biological flagging system. It literally means eat here.
So normally the Y grabs the bacteria, the handle sticks out, the macrophage sees a handle and eats the whole thing.
Enter Staphylococcus aureus with protein A. This protein is heavily embedded all over the bacterial cell wall, and it has a bizarre chemically specific affinity for the FC region, the handle of your antibodies.
Wait, so it grabs the antibody backwards?
Yes, he grabs the handle. So now the bacteria is completely coated in your antibodies, but they're all facing the wrong way. The grabber part of the antibody is facing outward, just waving at nothing.
And the handle is buried deep in the bacterial wall.
Precisely. So when that macrophage swims by, looking for a handle to grab, it doesn't see a target. It just sees the self part of the antibody facing out.
That's incredible.
It acts as a cloak of invisibility made entirely out of the enemy's own targeting flags.
That is almost indistinguishable from intelligence. It's not just a passive wall. It's an active camouflage system.
It effectively blinds the adaptive immune system.
Yeah.
You have to remember, S. aureus isn't thinking. It doesn't have a brain or a strategy room. This is just pure, ruthless evolution. Millions of years of blind trial and error resulting in the absolutely perfect defense mechanism.
And it's not even the only way they avoid being eaten. The outline mentions some strains have a capsule.
Right, a slime layer. Some strains produce a thick polysaccharide capsule on the very outside that makes them incredibly slippery. Imagine trying to grab a wet bar of soap in the shower.
It just shoots out of your hand.
That's exactly what a phagocyte experiences when it tries to engulf an encapsulated staph. The text actually notes that serotypes 5 and 8 are the ones causing most human infections.
Because their capsules are the best at slipping away.
Exactly. They are the most effective at preventing phagocytosis.
And then there's something called the clumping factor. Is that the same thing as the coagulase we talked about earlier?
No, and the text makes a very specific point to distinguish them. Coagulase is an enzyme that they secrete out into the environment around them. Clumping factor, on the other hand, is firmly attached directly to the cell wall itself.
What does it do?
It binds to fibrinogen and fibrin in your blood. This helps the bacteria clump together, hence the name, and adhere tightly to new hosts or tissues. It's basically like having industrial Velcro on the outside of the tank.
So just to recap the armor, we have a tank with a thick hull, Velcro to stick to things, a slime layer to make it slippery, and a camouflage system that holds the enemy's flags backwards.
It's formidable.
It really is. But a tank isn't actually dangerous unless it has weapons. Let's move to section 3, the arsenal, the enzymes.
This is where S. aureus really goes on the offensive. It produces a whole suite of enzymes designed specifically to modify the battlefield of your body. First up, we have catalase. Remember hydrogen peroxide?
Yeah, the bubbly stuff I put on cuts when I was a kid.
The very same. Now, why did your parents put it on cuts? To kill bacteria. But interestingly, your own immune cells also manufacture hydrogen peroxide as a chemical weapon. When a white blood cell actually manages to eat a bacterium, it dumps peroxide on it inside the cell to dissolve it.
It literally bleaches them to death.
Basically, yes. But staph produces catalase. This is an enzyme that rapidly converts hydrogen peroxide into harmless water and oxygen gas.
So it turns the immune system's poison into water. That explains the bubbles when I pour it on a cut. It's the bacteria neutralizing the attack in real time.
Yeah, exactly. That bubbling you see is the oxygen gas being rapidly released. It's quite literally the sound of the bacteria surviving. And if you are in the lab and you want to know if a random colony is staph or strep, you just drop some peroxide on it.
If it bubbles, it's staph.
Right. Strep doesn't have this shield, so it just dies quietly.
Next on the list is coagulase, which we touched on earlier. It clots plasma. But why does it want to create a clot inside my body?
The prevailing theory is that by clotting the plasma around itself, the bacteria coats its entire colony in a layer of fibrin. This creates a physical wall or a shell. It effectively builds a bunker around the colony.
To hide.
Yes, it protects it from immune cells and further inhibits phagocytosis. It walls itself off from the war going on outside.
But wait, if it walls itself off, how does it spread? I mean, if you're in a bunker, you're safe, but you can't exactly conquer new territory.
That is a great observation, and that's exactly where the next enzyme comes in, staphylococci. This enzyme does the exact opposite. It dissolves clots. It's the functional opposite of coagulase.
Wait, why would it produce both? That seems totally counterproductive. Build a wall just to tear it down.
It's all about timing and adaptability. It uses coagulase to build a bunker and hide when it is first establishing the infection and needs to multiply safely. Then, when the coast is clear, or maybe when they have run out of local nutrients and need to move, they secrete staphylokinase to break down the door, a process called fibrinolysis, and they escape into the bloodstream.
That is incredibly devious. It's literally controlling the terrain of the battlefield.
It dictates the terms of the engagement. And if they need to move through your solid tissues, not just the blood, they use hyaluronidase. We refer to this in medicine as the spreading factor.
What does that one do?
It breaks down hyaluronic acid.
I've heard of that in skincare.
Right, it's very popular in cosmetics. Hyaluronic acid is essentially the biological glue that holds your connective tissue together. It's the cement between your cells. Hyaluronidase dissolves that cement.
So it just melts the tissue.
Exactly. It's like taking a machete and clearing a path through the dense jungle of your body, allowing the infection to spread rapidly from a localized little spot to a much wider area.
And finally, the most famous enzyme of all in the medical world, beta-lactamase.
The ultimate shield against penicillin.
How does it work?
Penicillin works by attacking the bacterial cell wall. Specifically, a part of the antibiotic molecule called the beta-lactam ring binds to the proteins that build the wall, stopping construction. Beta-lactamase is an enzyme the bacteria produces that literally cuts the penicillin molecule in half before it can even reach the wall.
It just breaks the sword before it strikes?
It breaks the beta-lactam ring, neutralizing the drug entirely. This is exactly why basic penicillin hasn't reliably worked on staph infections since the 1950s. The bacteria learned to dismantle the weapon.
So it can dismantle our drugs, dissolve our connective tissues, wall itself off in a bunker, and neutralize our immune system's bleach.
And we haven't even gotten to the offensive weapons yet.
Right, because we aren't done. Section 4, chemical warfare, the toxins. This is where the source material gets truly graphic.
Yes. While the enzymes we just talked about help the bacteria survive and spread, toxins are designed to damage the host directly. Let's start with the hemolysis, specifically alpha toxin.
This is the pore-forming toxin, right?
Correct. It punches holes. Yeah. It acts directly on the cell membranes of red blood cells and platelets. It physically inserts itself into the lipid membrane and forms a ring, creating an open pore.
And then what happens?
The cell contents just leak out, and the cell dies. It destroys the very blood it lives in to get at the nutrients inside, specifically iron, which it needs to grow.
And then there's something called the Pantin-Valentine leukocidin that is quite a mouthful. Let's just call it PVL.
PVL is a profoundly nasty piece of work. It's what we call a two-component toxin, a leukocidin, that specifically and exclusively targets white blood cells, the leukocytes.
So alpha toxin kills the red blood cells to get food, and PVL kills the soldiers sent to fight the infection.
Exactly. It creates pores in their membranes and kills them on contact. PVL is particularly prevalent in community-associated MRSA or CA-MRSA. It leads to massive local inflammation and necrosis, which is dead, rotting tissue.
That sounds awful.
It is. When you see a really nasty necrotic skin infection in an otherwise healthy young person, like an athlete or a military recruit, PVL is very often the culprit. It essentially turns the immune system's arrival into a graveyard of dead white blood cells.
Now, speaking of skin, we have to talk about the exfoliative toxins. The outline calls these the skin melters.
These are proteases, enzymes that actively digest protein. But they are incredibly specific in their target. They dissolve the mucopolysaccharide matrix of the epidermis.
In plain English?
They dissolve the exact glue that holds your top skin layers to the deeper layers. Specifically, they target a protein called desmoglein 1, which literally anchors the skin cells to each other. When this protein is cut by the toxin, the skin layers just slide apart.
This causes staphylococcal scalded skin syndrome, or SSSS?
Yes, the skin literally separates and peels off in large sheets, looking exactly as if the patient has been severely burned with boiling water.
Who does this usually affect?
It typically happens in very young children and infants. Their kidneys simply can't clear the toxin from their blood fast enough. It's absolutely terrifying for a parent to see.
And the text mentions there are two types, toxin A and toxin B. Does it matter which one it is?
Not really to the patient. Toxin A is encoded on the chromosome and is heat stable. Toxin B is on a plasmid, which is a mobile piece of DNA, and it's heat labile, meaning it's destroyed by heat. But the clinical effect is exactly the same. Interestingly, they are both superantigens.
Let's pause on that word, superantigen, because we see this term again with the next toxin, TSST1. What exactly is a superantigen? How is it different from a normal, regular antigen?
This is a crucial concept in immunology. Normally, an antigen triggers a very specific, highly targeted immune response. Say your body sees a flu virus. Maybe 0.01% of your T cells recognize that specific virus shape and attack it. It's a sniper shot.
A precision strike.
Right. A superantigen, on the other hand, is like pulling the fire alarm in a crowded theater. It binds to the MHC class II molecules in the T cell receptors without needing a specific shape match. It bypasses the lock and key system entirely.
So what does that do?
It indiscriminately activates up to 20% of all your T cells at once.
20%? That sounds like a massive overreaction.
It is a catastrophic overreaction. All those millions of T cells simultaneously release cytokines. Cytokines are chemical signals that cause systemic inflammation. This sudden flood causes what we call a cytokine storm.
And what does that do to the body?
The body goes into profound shock. Sudden high fever, dangerously low blood pressure, a full body rash, and rapid multisystem organ failure.
This is toxic shock syndrome, or TSS.
Exactly. TSST1, the toxin we mentioned, is the prototype superantigen. This is the exact one associated with high absorbency tampon use in the 1980s, where the toxin would be produced in the vagina and enter the bloodstream. But it can also occur in regular wound infections.
So the bacteria stays in the wound, but the toxin goes everywhere.
Precisely. The bacteria might just be in a small cut on your leg, but the toxin floods your blood and causes systemic collapse.
And finally, rounding out the chemical warfare, we have the toxins that ruin the potato salad picnic. the enterotoxins.
Food poisoning. There are 15 distinct types of enterotoxins labeled A through P. And the crucial fact here, and if you take only one thing away for your personal kitchen safety, let it be this, is that these toxins are heat stable.
Okay, so let's say I cook a ham that got staph on it from someone's hands.
The heat of the oven will absolutely kill the bacteria. The staph is completely dead. But the toxin they produced, while sitting at room temperature, remains completely active. It easily resists boiling for 30 minutes. You can eat perfectly sterile, well-cooked food that is loaded with active toxin.
And what happens when I eat it?
It acts directly on neural receptors in your gut. These receptors send an immediate urgent signal up the vagus nerve directly to the vomiting center in your brain. It is essentially a rapid eject button.
So it's not an infection in my gut. The bacteria aren't multiplying inside my stomach.
No, it is strictly an intoxication. You aren't infected, you are poisoned. That's exactly why it hits so fast. Usually 1 to 8 hours after eating the food.
Wow, that's fast.
An actual bacterial infection takes days to incubate. This takes hours. And because it's a poison, it resolves quickly, usually in about 24 hours once your body clears it. There's no fever, just sudden, violent vomiting and nausea.
Okay, so we've covered the weapons. We've got pore punchers, skin melters, immune storm triggers, and vomit inducers. But a tank loaded with weapons isn't actually dangerous without a driver. This brings us to Section 5, the intelligence. The source material talks about something called the agrolocus. This strongly implies the bacteria make decisions.
They absolutely do make decisions, chemically speaking. This is a process called quorum sensing. The bacteria can actually sense their own population density. They're essentially counting how many of their brothers and sisters are present in the immediate area.
Like a roll call.
Sort of. They constantly secrete a tiny chemical signal into their environment, an auto-inducing peptide, or AIP. When the density of bacteria is low, the concentration of this AIP floating around is very low. The signal just drifts away.
So at this stage, the bacteria know they are alone or just a small squad?
Exactly. They know they don't have the numbers for a frontal assault.
So what's their strategy when they are few?
Stealth and attachment. The genetic programming focuses on expressing surface proteins, things like protein A for camouflage and coagulase to build a bunker. They want to stick to the tissue, hide from the immune system, and establish a quiet foothold without causing too much trouble or triggering an alarm. The strategy is, we're few, let's dig in.
But as they multiply and grow,
the population density increases, and naturally, the local level of that AIP signal rises. It starts to accumulate around them. Once it hits a very specific critical threshold, it begins binding to a receptor on the surface of the bacteria themselves. This binding event triggers the agar-regulatory system.
the accessory gene regulator, the switch flips.
Yes, the switch flips. It actively turns off the production of the attachment proteins and turns on the mass production of toxins, the hemolysins, the proteases, the superantigens.
The strategy becomes, we're many, let's attack.
Precisely. It radically switches from a colonization and hiding strategy to a full-blown invasion strategy. It creates a regulatory molecule called RNA3, which is the master switch for this whole operation. It downregulates the sticky hiding stuff and upregulates the killing stuff so they can destroy tissue and spread to new areas to get more nutrients.
That is terrifyingly smart. It ensures they don't waste precious energy making toxins until they have the sheer numbers to make it count. Because if just one bacterium released toxin, the immune system would spot the damage and wipe it out immediately.
Right, but if a billion of them do it at the exact same second,
It completely overwhelms the host. It's a coordinated, synchronized assault.
it is a microscopic blitzkrieg.
So what does this war actually look like on the human body? Let's move to Section 6, pathology. We start with the prototype lesion, which the book calls the furuncle.
Or, in plain English, the common boil. It almost always starts in a hair follicle or a sweat gland. The bacteria get in, start to multiply, and immediately use that coagulase enzyme to wall themselves off with a fibrin barrier. This causes intense, highly localized inflammation.
It gets red, hot, and swollen.
Yes, neutrophils, your frontline white blood cells, rush in to fight. They clash with the bacteria and die in massive numbers. The center of this walled-off area becomes necrotic. The tissue literally turns to liquid.
And that liquid is pus. Pus is essentially dead white blood cells and dead bacteria, right?
Yes, pus is the casualty of that microscopic war, and the body tries incredibly hard to fight it. But because of that thick fibrin wall, the bunker, your fresh immune cells and your antibodies have a really hard time getting inside to clear it out completely.
So what happens? It just keeps growing.
The abscess builds pressure and points in the direction of least resistance, which is usually upwards towards the skin surface, and eventually it ruptures and drains. And drainage is the absolute key to healing. Once the pus is physically out, the pressure drops, the bacteria are flushed away, and fresh granulation tissue can fill the void and heal the wound.
But if it doesn't drain outwards, it spreads inwards.
Yes, and that is where it gets dangerous. From that localized boil, it can invade the lymphatic system or enter the blood directly, causing bacteremia. And S. aureus in the bloodstream is a disaster because it can travel anywhere and seed other organs.
The source material lists some specific destinations. Let's go through them. Osteomyelitis.
That is an infection deep inside the bone. If a child comes into the ER with a sudden, severe bone infection, there's a 70% chance the culprit is S. aureus. It usually targets the metaphysis of long bones, the growing ends near the joints, like the knee.
That sounds incredibly painful.
It is excruciatingly painful and very hard to treat because most oral antibiotics don't penetrate solid bone tissue very well. It often requires weeks of 5E antibiotics.
What about pneumonia?
Staph causes a very severe, often necrotizing form of pneumonia. It doesn't just inflame the lungs, the toxins actively destroy the delicate lung tissue. And it can cause empyema, which is a massive collection of pus in the pleural space around the lungs.
Who gets that?
Interestingly, this very often follows a standard viral flu infection. You get influenza, your lungs' natural defenses drop, and staph, which might just be sitting in your nose, moves down into the lungs for the kill.
Wow. And then there's endocarditis.
This is one of the most universally feared complications. It's an infection of the heart valves themselves. Unlike some other bacteria that cause a very slow, subacute, smoldering endocarditis, S. aureus is acute and incredibly violent.
It moves fast.
It can literally destroy a healthy heart valve in a matter of days. The bacteria form these large, vegetation-like clumps on the leaflets of the valve, eating holes in them and causing sudden catastrophic heart failure.
We also talked earlier about the toxin-mediated diseases where there is no active infection. Food poisoning, toxic shock syndrome, scalded skin syndrome. We covered the mechanisms, but clinically, I want to distinguish between the rash of TSS and SSSS. They both cause skin peeling, right?
Good point. They do both cause peeling, but they look very different. In toxic shock syndrome, or TSS, the rash is scarlet cuneiform. That means it looks like a really bad diffuse sunburn all over the body. Then a week or two later, the skin peels desquamation, especially peeling off the palms of the hands and the soles of the feet.
And SSSS.
In scalded skin syndrome, the peeling happens immediately. The skin forms massive, fluid-filled blisters called bullae, and the top layer of skin peels off in large sheets at the slightest touch because that connection protein is dissolved. So both involve peeling, but SSS is superficial and blistery from the start, while TSS is a systemic shock with a full-body sunburn-like rash that peels later.
Got it. Now let's give a little screen time to the quiet cousins. Section 7, the coagulous-negative cousins. S. epidermidis. You mentioned earlier that it loves plastic.
It absolutely loves plastic. Intravenous catheters, prosthetic joints, artificial heart valves, and the way it survives on these lifeless surfaces is by producing a biofilm.
Explain biofilm for us. Is it just like a layer of slime?
It's much more than just slime. It's a complex, structured community. The bacteria secrete a thick, sticky polysaccharide matrix that glues them firmly to the plastic and to each other. This biofilm essentially acts like a microscopic force field.
So it sticks them to the implant.
Yes, and it protects them. It allows them to adhere tightly to the inert, smooth surface of the implant where human cells can't easily grab hold.
And antibiotics can't get through this force field.
Exactly. The biofilm physically protects the bacteria from drugs and from your white blood cells. The antibiotic you take might successfully kill the loose bacteria floating on the surface of the slime, but the ones buried deep inside the biofilm survive just fine.
So how do you cure it?
Very often, you simply can't cure these infections with antibiotics alone. The drugs just can't reach the core. You actually have to go in surgically and physically remove the infected implant. You have to take the hardware out.
That's a huge deal for a patient with a hip replacement. And here's the real kicker from the text. S. epidermidis is frequently resistant to methicillin.
Yes. Because it natively lives on our skin and we carry it into hospitals, it is constantly exposed to the many drugs we use in those environments. Through pure natural selection, about 75% of hospital strains of S. epidermidis are now highly resistant.
So even the supposedly weak, quiet cousin is essentially a superbug now.
It is. It's a massive problem in critical care.
What about S. saprophyticus, the other cousin?
This one is interesting primarily because of its very specific resistance to an older antibiotic called novobiocin. That's actually the key trick we use to identify in the lab. If a coagulase-negative staph is resistant to novobiocin, it's almost certainly saprophyticus.
And what does it do?
As we mentioned, it's the second most common cause of UTIs in young women, right after E. coli. It's not usually a hospital-acquired bug. It's very much a community bug.
Okay, we finally arrived at the heavy artillery. Section 8, the resistance crisis. This is the part of the deep dive that really keeps public health officials awake at night.
It absolutely should. The history of treating staph is basically a history of human triumph followed quickly by failure. When penicillin G was first introduced on a wide scale in the 1940s, it was hailed as a miracle. It completely wiped out staph infections. Doctors genuinely thought the war against this bacteria was over.
But the bacteria evolved.
Almost immediately, they acquired a plasmid, a circular piece of transferable DNA that contained the code for beta-lactamase, the enzyme we discussed that cuts the drug in half.
Right, so by the 1950s, standard penicillin was basically useless against most staph.
Exactly. So human scientists got back in the lab and created a new class of drugs, penicillinase-resistant penicillins. Things like methicillin, nafcillin, and oxacillin.
How are they different?
They were engineered specifically with a bulky physical side chain molecule attached to them. This bulky side chain physically blocked the beta-lactamase enzyme from getting close enough to cut the ring.
It's like putting a cage around the sword. You can't cut me, I'm armored.
Precisely. And it worked beautifully for a while. But then the bacteria pulled an entirely new trick. They developed into MRSA, methicillin-resistant Staphylococcus aureus. And this mechanism is profoundly different. It's not about cutting the drug anymore.
Please explain this carefully, because I think a lot of people get confused by MRSA. How does it work if it's not destroying the antibiotic?
To really grasp MRSA, we have to understand that this wasn't just a small random mutation in a single gene. This was the sudden acquisition of alien DNA.
Alien DNA? From where?
In a manner of speaking, Aureus didn't just tweak a gene it already had. It picked up a massive, entirely foreign chunk of genetic code called the MECA gene, most likely from a completely different, unknown species of bacteria. This gene is transported on a mobile genetic element called the SCC-MEC cassette.
It's literally like downloading a massive software patch from a completely different operating system.
That is exactly what it is. And this patch installs a brand new piece of hardware in the cell.
Yeah.
Earlier, we talked about penicillin binding proteins, or PBPs. These are the construction worker enzymes that physically build the peptidoglycan cell wall.
And penicillin kills staph by handcuffing these workers.
Right. The drug binds permanently to the PBP, paralyzing it, and the wall falls apart. Well, the MECA gene codes for a stunt double. It creates a completely new alternative construction worker, a new protein called PBP2A.
How is PBP2A different from the regular construction guys?
PBP2A is structurally distorted. Its active site, the molecular pocket where it grabs the building blocks, and where the penicillin tries to bind is closed off or shaped completely differently.
So methicillin is trying to get into that pocket to bind and paralyze it, but it physically doesn't fit.
Exactly. It has what we call an incredibly low affinity for all beta-lactam antibiotics.
So the drug is just floating around inside the cell looking for a place to latch on, but PBP2A just shrugs it off.
It doesn't even notice the drug is there. While all the native original PBPs are handcuffed and rendered entirely useless by the methicillin, this new PDT2A just keeps right on working. It takes over the entire job. It's a solo act. It builds the whole peptidoglycan wall all by itself.
Wow. It's like having a construction crew where the police show up and arrest everyone, but one guy just keeps laying bricks because he's wearing a disguise they don't recognize.
And that one guy is more than enough to keep the bacteria alive and replicating.
Yeah.
That's why beta-lactams, which includes penicillin, cephalosporins, carbapenems, all of them completely fail. They all absolutely rely on binding to that active site, and PBP2A simply won't let them in.
So it didn't even bother attacking the drug. It just changed the lock on the door so the key wouldn't work. That is genius and terrifying.
It renders almost an entire generation of our best drugs utterly useless.
Now, the text mentions there are different types of MRSA, hospital versus community.
Yes. HAMRSA, the hospital-associated kind, usually carries SCC-MEC types 1, 2, or 3. These are very large genetic cassettes, and because they are large, they often carry extra resistance genes for other non-beta-lactam antibiotics too.
Making them multi-drug resistant.
Exactly. They are the supertanks. CA-MRSA, or community-associated, usually carries a smaller cassette, SEC-MEG type 4Phi.
And CA-MRSA is the one that infects healthy athletes and kids, right?
Yes. Because the cassette is smaller, the bacteria reproduce faster. It's much more transmissible. It actually tends to be susceptible to some non-beta-lactam drugs like sulfa drugs or clindamycin, but it is highly virulent. It almost always carries that PVL toxin we talked about earlier.
The one that kills white blood cells and causes necrosis.
Right. It causes those severe, painful skin infections in perfectly healthy people, and it spreads rapidly in crowded places like locker rooms, gyms, and prisons.
So if methicillin and all the beta-lactams don't work, we have to use the big gun, vancomycin.
Vancomycin has been our absolute safety net for decades. It's a different class of drug, a glycopeptide. It works entirely differently. It doesn't try to bind to the construction workers. It binds directly to the cell wall building blocks themselves, preventing them from being linked together.
You're going to tell me resistance is appearing there too, aren't you?
Unfortunately, yes. We are seeing VISA and VRSA.
Let's break those down. What is VISA?
VISA stands for vancomycin intermediate S aureus. These bacteria haven't acquired a magical new resistance gene. Instead, they just drastically overproduce their wall. They build a massively thick, disorganized cell wall.
How does that help them?
It acts exactly like a sponge. It physically traps the large vancomycin molecules in the outer layers of the wall, so the drug gets stuck and can't reach the inner membrane where the actual wall synthesis is happening. It just absorbs the blow.
And VRSA?
VRFA, vancomycin-resistant S aureus, is the absolute nightmare scenario. In this case, the staph bacteria actually acquired a highly specialized resistance gene, the VAN-A gene, from a completely different genus of bacteria called Enterococcus.
Another interspecies gene swap.
Yes. This VAN-A gene literally changes the chemical sequence of the cell wall precursor. Normally, vancomycin binds to a very specific twin amino acid sequence, D-alanine-D-alanine.
Okay, D-aladila.
The VAN-A gene changes that target sequence to D-alanine-D-lactate.
It changes the shape of the binding site itself?
Exactly. It removes the exact hydrogen bond target the drug needs. So the vancomycin molecule literally cannot grab onto the cell wall. It floats right past. The bacteria is completely immune to our drug of last resort.
That is deeply concerning. Are we seeing a lot of this?
Fortunately, true VRSA isolates are still incredibly rare globally, mostly confined to specific geographic pockets, but the genetic mechanism exists, and the potential for it to spread is always there.
And there's one more resistance concept mentioned in the outline, tolerance.
Yes, tolerance is a much more subtle defense. The bacteria are technically inhibited by the drug. They stop growing and dividing, but they aren't actively killed by it.
What's the difference?
In a laboratory setting, there is a big difference between the minimum concentration needed to stop growth, the MIC, and the concentration needed to actually kill the cell, the MBC. In tolerance strains, the MBC is extremely high, much higher than you can safely achieve in a patient's blood.
Why don't they die?
It is often due to a lack of autolytic enzymes. Normally, when a wall-building drug damages a bacteria, the bacteria's own recycling enzymes go haywire and pop the cell. Tolerant bacteria lack these enzymes. They just go dormant, hunker down, and wait it out, but they don't pop.
And then when you stop taking the drug, they wake back up.
Exactly. This causes devastating clinical relapses, especially in deep infections like endocarditis, where you absolutely need the drug to kill 100% of the bugs because the immune system can't reach them.
So we are dealing with a bug that can actively dodge, block, or sleep through almost everything we throw at it. Which brings us to our final section, Section 9. Diagnosis, treatment, and prevention. How do we actually fight back against this?
Well, accurate diagnosis relies primarily on culture. We have to grow it to test it. We use blood agar to look for that hemolysis and mannitol salt agar to isolate it from other bugs. We can also use rapid latex agglutination tests that instantly detect protein A or the clumping factor on the surface.
And for MRSA specifically?
For MRSA, time is tissue. We often skip the growing phase and use molecular tests like PCR to look for the MECA gene directly in the sample. That gives a definitive yes or no answer very quickly, which is crucial for moving the patient to isolation.
And for actual treatment?
The first, most unbreakable rule of Staph Club is drainage.
Explain that. Why isn't a strong IV pill enough?
You have to think about the physical reality of the infection. Antibiotics travel exclusively through the blood vessels. A mature abscess or boil is a thick wall of fibrin enclosing a pool of dead tissue and pus, with absolutely no blood flow inside it. It's a dead zone.
So the drug literally can't physically reach the bacteria in the center?
It cannot. The concentration in the center will be 0. You must drain the pus mechanically. Surgeons have an ancient saying, ubi pus, ibi evacua.
Yeah.
Where there is pus, let it out. You have to open the bunker.
Ancient wisdom holds true.
Yeah.
And once it's drained, what drugs do we use?
It completely depends on the susceptibility testing. If it is MSSA, meaning it is methicillin sensitive, it doesn't have the MEK gene we use, nafcillin or oxacillin.
Wait, really? We don't just use vancomycin for everything just to be safe.
No, and that's a common misconception. Nafcillin and oxacillin are actually much better and faster at killing sensitive staph than vancomycin is. Vancomycin is a heavy, slow drug. You only use it if you have to. But if it is MRSA, we are forced to use vancomycin the 5.
Are there any alternatives to vancomycin if it doesn't work or the patient can't tolerate it?
Yes, thankfully. We have newer agents like daptomycin, which depolarizes the cell membrane, or linezolid, which stops protein synthesis. And there's a very fascinating newer drug called ceftaroline.
What's special about that one?
Ceftaroline is unique because it is a cephalosporin, which is a beta-lactam, but it was molecularly engineered specifically to bind tightly to PBP2A. It is the only beta-lactam that works on MRSA. It was designed to perfectly fit that altered lock we talked about earlier. It's a 5th-generation cephalosporin.
That's amazing. Finally, what about prevention? Can we just vaccinate against this and be done with it? We have vaccines for everything else.
We have certainly tried. There have been massive multi-million-dollar efforts to target the clumping factor or the capsule or the toxins, but so far, no human vaccine has ever been successful. Every major clinical trial has failed.
Why?
The organism is just too complex. It has too many redundant ways to evade the immune system. If you block protein A, it uses its capsule. If you block the capsule, it uses coagulase. It's too adaptable.
So it literally comes down to washing your hands.
It really does. The source of the infection is almost always human lesions or the respiratory tract, specifically the interior nares, just inside the nose. About 30% of healthy people carry it there right now. Transmission is primarily via direct contact with contaminated hands and fomites, which are inanimate objects like clothing or hospital bedding.
So hand washing is the best defense.
In hospitals, rigorous hand hygiene is the single most important control measure.
And if someone is a known carrier, like they have it in their nose?
We can actively treat carriers. We use an antibiotic ointment called intranasal mupirocin. You swab it up the nose, and it can eliminate the carrier state. We very often do this aggressively before major surgeries, like knee replacements, to reduce the risk of the patient infecting their own surgical wound.
So let's bring this all together. We've looked at a microscopic organism with a genuinely complex social life, a physical suit of armor made of peptidoglycan, a chemical arsenal that literally turns our own immune system against us, and the terrifying ability to genetically evolve around our best drugs in real time.
It is the perfect ultimate pathogen in many ways. It is a permanent companion of humanity. Most of us carry it peacefully. It's only when that delicate balance tips a break in the skin, a weakened immune system from the flu, or the introduction of a piece of plastic hardware that it strikes.
It's a really sobering thought. Understanding staph isn't just for doctors and microbiologists. It's a profound lesson in rapid evolution and the absolutely critical importance of basic hygiene.
Absolutely.
Well,
It constantly reminds us that we are not alone in our bodies. We are an ecosystem.
before we go, I want to leave you with a thought to mull over. We talked about VRSA staph acquiring the Van-A resistance gene from a completely different bacteria, Enterococcus. If S. aureus can swap genetic codes so easily, and given that we are rapidly running out of completely new antibiotic classes, what exactly does the landscape of surgery and medicine look like 20 years from now?
That is the big question.
Right. If we reach a point where we literally cannot stop a simple skin infection or protect a hip replacement, are we returning to the pre-antibiotic era where a simple scratch from a rosebush could mean death?
That is the exact question that defines the entire future of infectious disease research. It is a race against evolution.
Something to think about next time you're washing your hands. Thanks for joining us on this deep dive into the Staphylococci.
Stay safe out there.